The nucleotide is the basic building block of DNA and RNA, and it has a phosphate and a 5-carbon sugar along with one nitrogenous base: adenine, thymine, cytosine, or guanine. James Watson and Francis Crick constructed the DNA double helix model in 1953. This model depicts the spiral shape of the double-stranded DNA molecule. This molecule consists of a deoxyribose and phosphate backbone with base pairs bonded by hydrogen bonds. In these hydrogen bonds, adenine pairs with thymine and cytosine pairs with guanine due to the base pairing rule. The hydrogen bonds are individually weak and can be broken through raising the temperature. The anti-parallel nature of DNA is due to the strands running in opposite directions. The leading strand begins on the sugar's 5th carbon, moving from the 5' to 3' direction, and the lagging strand begins on the sugar's 3rd carbon, which is the 3' to 5' direction. DNA replication is semi-conservative, meaning it results in one old strand and one new strand. DNA replication begins with helicase unwinding and unzipping at the replication fork. Then, RNA primase synthesizes and attaches RNA primers. These primers start the process and are complementary to the given DNA sequence. Next, DNA polymerases add complementary nucleotides in one direction. The leading strand is continuous, but the lagging strand is added piece by piece, and those pieces are called Okazaki fragments. There are two types of DNA polymerase. DNA polymerase III extends the RNA primer, while DNA polymerase I digests the RNA primer and replaces it with DNA. The editors, nucleases, remove incorrect nucleotides when they are added. Finally, DNA ligase zips by adding phosphate in the remaining gaps of the phosphate sugar backbone.
DNA transcription is a process that involves transcribing genetic information from DNA to RNA. What are the differences between DNA and RNA? DNA contains sugar deoxyribose, whereas RNA contains sugar ribose. The difference between ribose and deoxyribose is that ribose has one more oxygen than deoxyribose. DNA is stable under alkaline conditions, which means having a pH greater than 7, while RNA is not stable. DNA is a double-stranded molecule, while RNA is single-stranded. DNA is responsible for storing genetic information, while RNA codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. DNA uses bases adenine, thymine, cytosine, and guanine. RNA, however, uses adenine, uracil, cytosine, and guanine. There are multiple types of RNA, such as mRNA, tRNA, and ribosomal RNA. The exons in transcription are the codons that are expressed. The introns are the codons that are not expressed. The location of transcription occurs in the nucleus, and only exons are used in creation of protein. Transcription of a gene begins in the nucleus when the double-stranded nucleotide sequences flanking the region of the gene open, revealing the single-stranded DNA sequence to be transcribed. Only one strand of DNA is used as a template for transcription, using the DNA-RNA complementary base pairing rule and RNA polymerase as the builder. An A in the DNA strand places a U on the growing RNA strand, and a T in DNA places an A on the growing RNA strand. The three major steps in the process of transcription are called initiation, elongation, and termination. Once the entire DNA sequence has been transcribed, RNA polymerase encounters the terminal sequence of the gene and disassociates from both the newly transcribed RNA and DNA. The entire RNA thus detaches, and in eukaryotes it is further modified before it matures and moves to the cytoplasm. The DNA then restores its double-stranded structure. After transcription, several modifications must occur before the mature mRNA leaves the nucleus. To facilitate the transport of mRNA to its final destination and prevent it from deteriorating while in transit, extra nucleotides called a cap and a poly-A tail are added to both ends of the mRNA. Another modification is the removal of the non-coding region that has been transcribed from the gene. A typical gene in the eukaryotic genomic structure has intervening non-coding regions called introns and coding regions called exons. The intron portion transcribed to mRNA must be removed, and the exon portions must be stitched together before the final mRNA product is usable.
Translation is the process of converting RNA into proteins. During translation, the mRNA moves from the nucleus into the cytoplasm. A ribosome consisting of rRNA, ribosomal RNA, and proteins attaches to the mRNA. The genetic code of the mRNA is broken up into a group of 3 nucleotides, each group that codes for a different amino acid. Each one of these groups is called a codon. The tRNA, or the transfer RNA, enters the ribosome at the A site. Each tRNA carries a different amino acid and also carries an anticodon, or the matching pair to a codon on the mRNA. The tRNA then moves on to the P site, while another tRNA enters at the A site and matches with the codon. The amino acids then form a polypeptide chain through the formation of peptide bonds. The carboxyl group of one amino acid reacts to the amino group of the other amino acid and forms a peptide bond. After the peptide bond is formed and the amino acid is added to the polypeptide chain, the tRNA then moves to the E site, where it is ejected from the ribosome without the amino acid. A new tRNA then enters at the A site, while the tRNA previously in the A site moves on to the P site. Another peptide bond is formed between the amino acids, and the polypeptide chain gets longer. The tRNA at the P site then moves on to E site and is ejected. tRNA at the A site moves on to the P site, and a new tRNA enters at the A site. This process continues until a stop codon is encountered. After the stop codon is encountered, the process terminates, and the mRNA and the polypeptide chain or protein is released into the cytoplasm.
There are a couple different kinds of mutations that can occur within the process of DNA replication, transcription, and translation. One of these possible mutations is the frameshift mutation, which is caused by an addition or deletion of nucleotides. This can result in a change of the reading phrase and result in a completely different translation and different reading of codons. As a result, there are different amino acids. This frameshift mutation is potentially the most harmful of all the mutations. There is also substitution mutation, which is also known as point mutation, in which one specific base is exchanged for another one. For example, if adenine was used instead of cytosine. And this results in a different codon and a specific different amino acid. However, the effects of this are much less subdued in comparison to the ones such as a frameshift mutation because of the fact that only one codon would be affected. There's also an inversion, which is essentially what happens when a part of a DNA sequence is reversed back to front instead of front to back. And this results in a completely different reading and a completely different codon and thus amino acids being resulted. This, however, is a very detrimental mutation, however not as horrible as frameshift mutation can be because of the fact that frameshift can change the entire sequence, while inversion only changes a specific part of it. Now there is also the least harmful, the silent mutation. A silent mutation is essentially a mutation that occurs in a non-coding region, where the DNA in itself is not being read. And as a result, a mutation that is mutated within specific non-coding regions are called silent mutations because of the fact that they do not affect the phenotype. Multiple triplets can code for the same amino acid, so if a base gets substituted for one that still makes the same amino acid, the phenotype won't change. This is also an example of a silent mutation.